Okay, we are going to talk about Chapter 20, Objective 1. Sorry, I had to get there. Chapter 20 is dealing with blood vessels, and Objective 1 is talking about the structure of the various blood vessels, arteries, veins, and capillaries. Just to remind you all, the difference between the pulmonary and systemic circuit. We talked about this back in Chapter 19. I told you all there was a lot of overlap between Chapter 19 and Chapter 20. So the pulmonary circuit is where the right ventricle sends blood to the lungs and it comes back to the left atrium. Whereas the systemic circuit, make that a different color, is going to take blood from the left ventricle, go to the body, and return it back to the right atrium. So here's your systemic circuit, and above is that pulmonary circuit, which is taking blood for gas exchange in the lungs. Now, there are three main types of blood vessels, and then we have some smaller vessels that have some different names. So this chart right here is showing you the diameter of the blood vessel. Now, diameter is the distance from one end to one end. Remember, radius goes from the middle outward. Diameter is the distance from the inside to the other side of the blood vessel. The thickness of the wall, how much material it's made up of. So in other words, how much of the endothelium does it have? How elastic is it? How much smooth muscle does it have? And how much fibrous tissue it has. We talked about how smooth muscle surrounds blood vessels to allow them to either constrict or dilate, right? To have the opening of the blood vessel get smaller, constrict, or dilate, have the smooth muscle relax, and the blood vessel will get larger in diameter. So knowing which blood vessels contain the most smooth muscle kind of helps with that. And then how elastic are they? What's the elasticity? So in other words, if blood is moving through it, how available is that blood vessel going to be to allow for increased pressure within that blood vessel? Some not so much. Others are going to allow for a lot of elasticity. And then endothelium, that's just the inside lining of the blood vessel. And you can see pretty much they all have the same amount of endothelium. That's the same for all blood vessels. The other thing I want you to notice is arteries. You have to be careful on this one because that artery, the unit, watch the units, that's millimeters. So this right here is micrometers, which is 1,000 more times smaller than a millimeter. So I do wish that they would have put this all in the same unit. That would have been a little bit nicer. So this would really be 0.04, if I did that right, 0.004 millimeters as far as the diameter. So again, the diameter of I went the wrong way with my decimal point. Sorry. How about that? Sorry, went the wrong way. So the diameter of an artery is going to be, just looking at the consistent units there, diameter of an artery is going to be 400 micrometers, an arterial 30 micrometers, capillary 8 micrometers. So the diameter gets smaller as we go. And then the capillary, it's going to have the smallest diameter. And then as we go out from the capillary, the diameter is going to start getting larger again. So the venule is going to be 20 micrometers. And then for the vein, that would be, moving the decimal point, 500 micrometers. And you can see that the vein has a larger diameter than an artery does, which you can kind of see that the vein actually has the largest diameter blood vessel. So blood vessels are going to start out large. They're going to get small as they turn, blood moves into the capillaries. Capillaries have the smallest diameter. And then as blood leaves the capillary, the diameter of the blood vessel is going to get larger and larger and larger. And it just so happens that diameter of the veins is larger than an artery. So take a look at the vein in the histology slide versus the artery in the histology slide. The diameter, the inner diameter of the artery is very nice and round, whereas the vein is a little bit more oblong in structure and a little bit bigger too. So again, you can see the veins in this histology image, how they're larger and kind of not nice and circular shaped. Whereas with the artery, you can see that it's pretty much a nice little circle and has a very nice wall that's very visible. So arteries carry blood away from the heart. That's 100% true all of the time. What I told you all in the lab, what's not true, is arteries always carry oxygenated blood. That's not true. We know that the pulmonary artery is deoxygenated. So again, arteries, while they always take blood away from the heart, most of the time they're oxygenated, but not 100%. Now, arteries can be, they're going to have some elastic properties, which means they're going to have some give to them when blood is being pumped through them. So that way they don't burst, right? So they'll have a little bit of elasticity in them. They're also going to be fairly muscular, meaning they're going to have quite a bit of smooth muscle surrounding them so they can control the diameter. The walls are very thick. You can see the thick elastic wall in the artery compared to the thin wall, non-elastic, in a vein. Small lumen, meaning the opening, the diameter that the blood is going to be passing through is very small in comparison to a vein, which has a very large diameter opening. Why is that? Arteries have to be very reinforced because there's, again, a lot of pressure in the artery. Veins, they've already passed through a capillary. There's not a whole lot of pressure left in a vein. So they're okay to have a little bit thinner of a wall, less elasticity, and a larger lumen. They're just carrying basically blood that's not under a whole lot of pressure. That's why when you determine blood pressure, you're looking at blood pressure of an artery, not of a vein. If you increase the pressure, you're increasing the resistance. Just kind of FYI, pressure is directly related to resistance. Arterioles, they're just smaller arteries working its way towards a capillary. So they're just smaller and smaller arteries. Capillaries, they're actually the most common. So you're going to have capillaries everywhere. This is where the magic happens, so to speak. This is where gas exchange occurs between the interstitial space and the capillary. So this is going to occur in the lungs, where the pulmonary capillaries are going to pick up the oxygen from the alveoli of the lungs and drop off that carbon dioxide waste to be removed. And then in the tissues, the reverse is going to happen. The capillary is going to drop off the oxygen to the tissues and pick up the carbon dioxide waste from the tissues and carry it onto the lungs for removal. Capillaries have the thinnest walls. Makes sense if you think about it. These thin walls allow for materials to transfer back and forth. And blood is going to travel very, very slowly. So we've gone from a fairly large blood vessel, an artery, and we've gotten that narrower and narrower and narrower until we finally reach a capillary. If you think about maybe going down a road or something like that, maybe one of the ones in Dallas, like 635, and they're flying down 635. And I don't know about you, but they like never do that. But anyway, they're going pretty fast down 635. It's 5 lanes on both sides. Everybody's happy. And then as you get closer to the I-30 exit, you lose lanes. And what's happening is the lanes are lessening as you get closer to your exit. Traffic's going slower. So it's kind of the same principle. When you're in a 1-lane road, you're going real slow. And that's what's happening in a capillary. These blood is traveling very slowly. A single cell at a time can only pass through the capillary. So they're very narrow. Blood is traveling slowly. Now, the purpose, we kind of like the blood traveling slowly in a capillary. That allows for maximum exchange of material. If it was traveling so fast, you wouldn't be able to do any exchange. So slow is good in a capillary. Venules, they're going to be, as blood passes out of the capillary, they're going to enter into veins, specifically small veins, which are called venules. And then those venules are going to merge together to form larger and larger blood vessels called veins. When we look at veins, they're going to bring blood towards the heart every single time. Most veins are deoxygenated, but pulmonary vein, for example, carries oxygenated blood to the heart. So we can't say that veins always have deoxygenated blood. That's not a 100% true statement. The fact that they always bring blood to the heart, that's true. The walls of the vein are very thin, like we see in the picture up here. Very large lumen, very large inner diameter opening there. So that means we don't have a lot of pressure and we don't have a lot of resistance. So blood flows fairly easily through a vein. They're not very elastic. You don't need them to be elastic. They're under no pressure. So it's okay that they're not elastic. And they don't have a whole lot of smooth muscle. So the purpose of smooth muscle, again, is to constrict or dilate blood vessels. We're not really trying to constrict and dilate veins. We're trying to constrict and dilate arteries prior to the capillary gas exchange location. We really aren't trying to constrict blood flow in a vein. We want that to flow freely back to the heart. And then, believe it or not, veins have valves. You can kind of see the valves in the picture here. Not very well. I'll show you a better picture of that. But veins actually have valves. And what is the purpose of a valve? I don't care if it's in a heart. I don't care if it's in a vein. I don't care if it's in the lymphatic system. The purpose of valves is the same. It is to prevent backflow of the material. So it's to prevent blood from going backwards in the vein, in this case, because we're talking about veins. Now, the endothelium. This endothelium is made up of simple squamous cells. Now, if you remember simple squamous, that was a single layer of flat-shaped cells. So if you look at the endothelial cells that line the inside of all blood vessels, whether it be an artery, a vein, or a capillary, the endothelium, this simple squamous layer of simple squamous cells on the inside lining, is the same. The other thing is you're also going to see this endothelium, this layer of simple squamous epithelial cells, line the inside lining of the heart. That was called the endocardium. Same identical cells. And this is continuous. So the endothelium of the heart is continuous with the endothelium of the blood vessels, which makes sense, seeing as how blood vessels come off the heart. That kind of makes sense. Capillaries, pretty much if you look at the structure of a capillary, the only thing you're really going to see are endothelial cells. There's really not much else to the capillary. They have these extra pericapillary cells. Peri means around. That's a cell that's around the endothelial cells. You can kind of see some pericapillary cells in the purple image over here on the bottom right hand of your screen. Those little purple things, those are pericapillary cells. You also have them over here. They're represented in purple. They could be fibroblasts. They could be macrophages. They could be smooth muscle cells. So basically, they're just cells that are pretty much on the outside of the endothelial cells. Now, again, red blood cells are going to flow pretty much single file. There is a basement membrane, which is a thin layer of connective tissue that surrounds the entire capillary. Again, what have we not talked about? What organ, what structure have we not talked about that doesn't have a thin layer of connective tissue that surrounds the outside of it? Now, right now I can't think of one off the top of my head. Okay, so there are actually 3 different types of capillaries. You have what's called continuous, fenestrated, and sinusoidal. So a continuous capillary, notice they all have the basic same structures, but as you go from continuous to sinusoidal, they become more porous. Okay, allowing material, if they're more porous, they're going to allow material to easily flow. So continuous capillaries, there's no gaps in that. Those epithelial cells are really smack dab right up next to each other. There is no space for anything to really get through. If you all remember when we talked about the blood-brain barrier, which was preventing material from going from the capillary, from the blood into the brain. And we said that astrocytes were the big factor in that. Another factor was the fact that these capillaries that are found there are of the continuous type. So the capillaries are also not allowing a whole lot of transfer between the blood and the brain. Again, sometimes that's a good thing. Sometimes that's not a good thing. If you're trying to get medicine to the brain, that's really not a good thing. They're not very permeable to substances. Things can't get in or get out because of the no gaps within the epithelial tissue. Okay, the fenestrated capillaries, they contain little holes called fenestrations. That's why they're fenestrated capillaries. The word fenestra means windows. I don't know what language that is. So the plasma membrane of the capillary is very porous, meaning a lot of things can move back and forth. Not a lot, but a lot more than in the continuous capillary for sure. So you're going to find these capillaries where you need a lot of exchange going on. The sinusoidal capillaries, these are larger in diameter. They have a lot of missing components as far as a lot of missing parts and pieces there. These are allowing huge chunks of material to pass from one place to another. So these would be things like you're actually passing whole cells through these capillaries. Endocrine glands, hormones. Some hormones are pretty darn big. Remember, hormones are going to be in the blood supply until they reach their target. Well, if the target is next to a sinusoidal capillary, that's going to ensure that hormone has the ability to exit the capillary and get to the target cell. Lymph nodes, very porous. Bone marrow, you're looking at large molecules that need to cross, such as reticulocytes, the precursor to a red blood cell. So these have to be very large. The pores have to be very large to allow the passage of larger structures. Now, with capillaries, they supply tissues with oxygen. However, we don't want all tissues to get all the oxygen all the time. If you're in sympathetic stimulation, fight or flight mode, your digestive system is kind of taken offline, so to speak, for the time being. You're trying to run from a bear. You're not trying to digest that hamburger. So we need blood flow diverted to the areas that you need, such as skeletal muscle, such as the brain. We need blood flow really going to those areas, not so much to the stomach. So what do? There are ways to seal off capillaries. Now, we can't just shut off a whole capillary because that means blood flow would never enter the vein and you'd get a stoppage right there. So you have what's called a meta-arterial, which allows blood to flow through the capillary without actually undergoing a lot of gas exchange. There's going to be a little bit of gas exchange, but not to the level that you would have if you opened up the capillary for blood to flow through. So you have got a shunt, which is that meta-arterial. It's a thoroughfare channel that allows blood to literally go from the artery straight to the vein without a lot of exchange going on. So this is a bypass, so to speak, of the capillary. Now you can see in the upper picture up here, when the capillary is open, you can see blood is going to be all in those capillaries. So what is allowing it to either not go through the capillary bed or to go through the capillary bed? So blood is always going to go through that meta-arterial, that thoroughfare channel. That's always going to be open. But what's closing it are these precapillary sphincters. Blood is regulated by these precapillary sphincters, and you can kind of see them here in this picture. Notice they're only on the artery side. You don't see them on the venous side because you don't need them on the venous side after they've already gotten over there. So you've got this tissue, smooth muscle, that's going to shut that blood vessel down, prevent blood going into that capillary, so it's just going to pass right on through. And again, an example of this would be, a rough example would be blood flow being shunted from the digestive system because it's not needed there when you're in fight or flight mode. Arteriovenous anastomosis. So this is another shunt. This is another way to get blood from entering into a capillary bed. So it's going to flow from an arterial into a venule without passing through the capillary. So you can see right here, you have a little shunt right there. So if it passes from the arterial to the venule, it's not going through that capillary bed. And again, you're going to have sphincters to block that capillary bed off to allow blood to go right from the arterial to the venule. So where are you going to have this? Where are some places you would see this anastomosis? An anastomosis, again, is when you have two blood vessels that are bypassing the capillary, two blood vessels that are connected together, not really allowing for gas exchange. So you're going to find them in the foot and the hand, in the terminal digits, and nail beds. So this is where you're going to have that. Part of it is to help regulate body temperature. So remember, when you're hot, you would open up these anastomoses, so you would not shunt it off. You would allow blood to flow through the capillary bed, and then do the gas exchange and then into the venule when it's warm, because you're trying to get excess heat out of the body. So in warm conditions, you would open up that capillary bed. When it's cold, you would shut that off and allow blood to just flow from the arterial into the anastomosis into the venule to conserve heat when it's cold. So you would block off that capillary bed. Now, like we've said earlier, you've got some different layers of the blood vessels here. So the names of them are the tunica intima, that's the inside green right there, the very inner layer. The tunica media, that's the middle layer. And then the tunica adventitia, which is the outer layer. So except for capillaries, you're going to see these three layers. Why not capillaries? Well, it's only made up of smooth, a simple simple squamous endothelial cells. So there is no room for all this other stuff. So it's just one thin layer of cells. So from the lumen to the outer wall, the layers are, again, tunica intima, tunica media, and tunica adventitia, also known as tunica externa. So the tunica intima, this is pretty much the endothelium, the simple squamous endothelial cells. A thin layer of maybe some connective tissue connecting everything together there, holding it together. And this is also where you're going to have some elastic fibers, potential elastic fibers that could be a little porous. Now, the tunica media, and you can see the tunica intima right here, which is those endothelial cells. And that's also the opening of the blood vessel, right? The lumen. Now, the tunica media, this is really the one that we've been talking about without talking about it in A&P1 when we discussed smooth muscle. So we said smooth muscle wraps around the blood vessels to either allow them to constrict or dilate. Specifically, it's in the middle layer. It's in the tunica media. So you do need to know that smooth muscle cells are found in the tunica media layer. This is going to regulate blood flow through the vessel, which we've said before, vasoconstriction versus vasodilation. And the tunica adventitia, this is just connective tissue here. We're really not going to spend a lot of time with it. This is going to vary with blood vessel type, so not all are created equal. Arteries are going to have more than, let's say, veins are going to have. Okay, now veins are going to be, you got different sizes, just like you have with arteries, small, medium, and large. Venules are going to be smaller in size. Veins are going to be the larger ones. Interestingly enough, larger veins contain valves. So the larger the vein, the more opportunity there is for maybe to find valves in that. And the purpose is to prevent the blood from backflowing, to make sure that blood is moving in a forward direction towards the heart. We're trying to get it into either the inferior or superior vena cava. We don't want blood going backwards. The other thing you can notice is the smooth muscle in the tunica media layer, a lot more smooth muscle around the artery than around the vein. Makes sense. So it's got a lot more of the tunica media. Now, one of the things that can happen is, what if those valves don't work properly? This is where you run into someone that might have varicose veins, typically found in the lower limbs. This is a pretty dramatic picture here on the right. but it gives you an idea of what this looks like. Defective valves, so you can see they're calling them incompetent valves. They're allowing the blood flow to not move in one direction. So it's allowing backflow. Now, veins are typically in the lower body bringing blood up to the heart. So if you're allowing backflow, this blood is going to pool in the lower extremities, and not be able to move towards the heart like it should. When that happens, you end up getting this kind of distension, this type of look in the veins at this point. So they become twisted, they become visible on the surface, and this is creating an issue where a clot could form, and that's not a good thing. And this is also it can be very painful as well. Should have probably warned you there was a real picture coming up here. So phlebitis is inflammation of the vein. So why would the vein become inflamed? Well, the inside could be irritated, and that's going to cause the cells on the inside to kind of have a rough spot. Anytime you've got a rough spot rather than smooth through the blood vessels, that's going to create an opportunity for platelets, remember those little guys there, to start clumping together and adhere, and that's going to cause inflammation. That is also going to result in a clot forming in that area, and it can prevent complete blood flow to that area, which can result in gangrene, which is death of the tissue, which you can see in this person's fingers over there, due to a reduction in the blood supply. This person is probably going to have to get some fingers amputated. I mean, once you've got tissue death at that level, it's kind of hard to get that back. You're going to see this in diabetics. That's one place you're going to see a lot of this stuff. Deep vein thrombosis, DVTs. Y'all have probably seen commercials on this. So a deep vein thrombosis, a thrombosis is a clot, is going to be in a deep vein of the lower body. Could be leg, could be pelvis, where it's either going to partially or completely block blood flow. Now, keep in mind, these veins are bringing blood back towards the heart. That's the idea. So even though they're red, they probably should be blue. Now, if a part of that clot breaks off and starts to travel through the circulatory system, it is then called an embolus. So a embolus is one where part of that thrombosis, that deep vein thrombosis, breaks off and starts traveling, and it's going to the heart. And that heart has the pulmonary circuit, so it could go into the lungs. And if that clot ends up entering the lungs, or it could even enter the brain, if it does that, it's going to block blood flow to that area, preventing a disruption of oxygen. And you certainly don't want that happening in your lung or in your brain or in the coronary arteries of your heart. Arteriosclerosis. This happens in arteries. And you can see progression of arteriosclerosis as time progresses in that bottom picture there. Another thing, it's typically called hardening of the arteries, and you can see how that artery looks like it's getting worse and worse. It has less elasticity because it's hardening, so it's not going to be as elastic. And remember, arteries need to be elastic because of the intense pressure they can be under from the blood pumping through them. If you make the arteries more hard, you're increasing the resistance of what's in the artery, creating it more difficult to push blood through. So we talked about increased resistance back in chapter 17. So this is bottom line. You're not going to get good circulation. Areas that need oxygenated blood are not going to get them because you now have arteries that are creating the problem, and that's going to cause increased work on the heart. Now, a specific type of arteriosclerosis is called atherosclerosis, and this is the type of hardening of the arteries that affects those coronary arteries, those specifically supplying the heart with oxygenated blood. And you can see the plaque build up there in that coronary artery. It's called atherosclerosis, again, a type of arteriosclerosis, and it's going to prevent blood flow through those coronary arteries. Okay, and that's it for Chapter 20, Objective 1. I'll find the right button in a minute. There we go.